Hello, this is Alex, your Lifeline leader, and this is what appears to be Lifeline Online. This week, we will be discussing mitosis. We are on week 10 of Lifeline, and there are 2 PPP points associated with this lecture today. Please remember that we are no longer meeting in person, so there is no signing sheet anymore. Previously, you used to get these PPP points just by signing the signing sheet. Given that that is not a possibility any longer, there are going to be weekly quizzes due on the Saturday of that week at 11:59. This means that this Saturday, if you want to get those 2 PPP points, you have to do the quiz on Canvas. Okay, it is a very short quiz. It is 5 questions, and you will get the points by doing the quiz. There is no way for me to tell if you watch the lecture unless you do the quiz. Okay? Moving on. A couple of reminders. This Saturday at 11:59, you also have Fact or Fiction due if you are in Group 6. So there is a lot of things due this Saturday. In addition to that, everyone has the presentation outline. So everyone should already be working on these outlines, please. Remember that only one person per group is required to submit the outline. Make sure that you pick a person, and more importantly, that you keep asking this person if the outline has already been submitted. Because if that person does not submit the outline for your group and there is no outline for your group, then I have no choice but to take off the 2 points for everybody. Furthermore, project presentations have not been changed. The dates for the project presentations are still the same. They will be due on week 12, the day of our meeting. So this would be on a Saturday. The Tuesday of week 12 is when you would have the project due. Alright? Now, obviously, the project can no longer be in person. And because of this, you will have to submit it online. All the instructions for this are already on the announcement that I made this week. So please give it a look and make sure that you understand everything that's on that announcement. Of course, I'm always reachable by email if you have any more questions. And then week 10 extra credit quiz is going to be due on next week, the Saturday of next week, and it's 2 points. It is going to be based on the materials that we will discuss here today. So let's get into mitosis, okay? There are a couple of words and terms that you must understand if you are going to understand mitosis properly. The first of those words is ploidy. The number of sets of chromosomes in an organism. That is what ploidy means. And ploidy can have several states. For instance, there is the haploid state, where an organism has only unpaired chromosomes, which consist of one complete set of DNA. So one complete set of DNA is the same as haploid. When organisms have 2 sets of DNA, then that is a diploid organism, because it has paired chromosomes. We will get more into these terms as we move forward. But for now, understand that having 2 sets of DNA means that that organism is diploid. In the same fashion, we can have a triploid organism, a tetraploid organism, a pentaploid organism. But in this class, and to be fair, pretty much in any other class in college, we are going to stick with haploid and diploid organisms. They are by far the most common, and that's all we are going to need for this class. Here, you can see the haploid representation. You can see 1 chromosome, 2 chromosomes, 3 chromosomes. But this doesn't mean that this organism is triploid because it has 3 chromosomes. The number of chromosomes does not matter. What we are looking at is at the number of sets. So here we have 1 set of 3 chromosomes. In this side, we have 2 sets of 3 chromosomes. Okay? We have 1 set. We have chromosome 1, we have chromosome 2, and we have chromosome 3. This is one set, and this is the other set. If we talk about us humans, we are diploid organisms. We are diploid because during fertilization, we get one set of chromosomes from our mother, and the other set, we get it from our father. Now let's look at the difference between chromosomes and chromatin. It is a little bit counterintuitive because I do not want you to think that chromosomes are one thing and chromatin is a completely different thing. In fact, they are both made up of the same genetic material, okay? They are both made up of the same DNA. The difference is that chromosomes are tightly packed DNA that can be found during replication. And this is when you will have chromosomes. During replication, alright? When the cell is turning into two cells. That is when you're going to see chromosomes. When that is not happening, when the cell is not dividing, we are in a process that's called interphase. That is basically any time that the cell is not actively in mitosis. That is when you have chromatin. Chromatin is unwound DNA that can be found during the interphase. So it looks like a soup of DNA, if you will. There is no shape to it. It is the whole double helix of DNA completely all over the place. Alright? Whereas chromosomes is when it gets a little bit more packed, a little bit more organized, and it gets a shape. So now let's look at the chromosomes themselves. Homologous chromosomes. A pair of chromosomes, one from maternal side and the other from paternal side, that have the same types of genes in the same location. That is homologous chromosomes, okay? Looking at the image, I want you to notice something. We are saying here, this is a diploid organism, as you can already tell. We have one set and we have another set. These are homologous chromosomes, as you can see by the genes that they have. Both of these have the same genes. However, not both of these are completely exactly the same. They are not. That is because this one chromosome over here can be said to come from mom, while this other one over here can come from dad. And this is very easy to understand. If you think about it, your mom does not look exactly as your dad. That is because we have the same genes. We all have a gene that codes for hair color. We all have a gene that codes for eye color. We all have a gene that codes for 2 ears. But those are different, and that is where alleles come into place. Each chromosome has alleles that code for different versions of the same gene. For instance, the allele for eye color, let's say it's this, in your mom could be for brown. Whereas in your dad, the same allele for eye color, the same gene for eye color, could code for something else, like hazel, for example. Okay? The two identical copies of the same chromosome connected at the centromere are known as chromatids. So first, let's wind up a little bit. What is a centromere? A centromere is just the area where these are attached. Now, let's establish something and make it clear. When we talk about this one over here, and this one over here, they are completely exactly the same. These are copies of each other. They are both from dad, for example. Both of these are both from mom. So yes, they are completely exactly the same. That is why they are called one duplicated chromosome. Again, the both of these, when you look at them together, they are homologous pair of chromosomes. Homologous. That does not mean that they are exactly the same. It means that they have the same genes, but it does not mean that they are exactly the same because they can have different alleles. Okay? As I was saying, the centromere is the bond between the exact two copies of DNA that ensures that chromosomes will function as one working unit. This is the centromere region right here. And that's all it is. It is a region. It is not a structure. There is nothing there. Well, actually, there is something there. There is the kinetochore. That is what physically holds both chromatids together. But the centromere is not a structure. It is the region where both of these are attached. The kinetochore is a structure. It is a protein that is associated with the centromere region and are important during cell division. We will see why. But that's the kinetochore. This right here is the kinetochore. And again, it is a structure, unlike the centromere, which is just a region. One strand of a duplicated chromosome is called a chromatid. So this is one strand. This right here, a chromatid. This right here, another chromatid. Alright? They are identical to each other. They really are. The chromatids are exact copies of each other, just what I was telling you, correct? Now let's take a closer look at us humans. The human karyotype. This picture is a human karyotype representing 23 pairs of chromosomes, which is what we all humans have. All of us who are healthy humans, okay? 22 pairs of autosomes and 1 pair of sex-determining chromosomes. So every pair of chromosomes that is not a sex chromosome, this right here, is an autosome. That is all it means. The autosomes have every other gene that has nothing to do with sex. Alright? This is where you would find the genes for your hair color. This is where you would find the genes for your height. The genes for your eye color, for your teeth, all of it. Whereas in here, in the sex chromosomes, this is where we are going to determine the sex of the individual. And there are a few other genes that are also associated with these. We are probably going to leave this until we get to the genetics unit. That is unit 4. Chromosomes pair up based on the size and centromere position of each chromosome. Here you see a total of 23 homologous pairs, and, as we already know, a pair has 2 chromosomes. So in total, there are 46 chromosomes in each cell of the human body, except for gametes. And we're going to leave that right there until we talk about meiosis next week. Let's look at the cell cycle. This cycle is composed of interphase, mitotic division, and cytokinesis. Cytokinesis is when the cell separates, when it divides into 2 new cells. As I told you before, interphase is any time that the cell is not actively engaging in mitosis. So here you see mitosis with the cytokinesis, and then everything else, all the gray part, is interphase. And yes, there are some stages of interphase as well. But understand that interphase is all the other time that the cell is alive when it's not actively engaging in mitosis. So interphase comes before each nuclear division and after each nuclear division, okay? Takes up most of the time during the cell. And there are 3 phases, as we saw in the previous picture. There is the G1 phase, during which the cell is growing, is doing its daily functions, everything that it needs to do to stay alive and grow. During the S phase, DNA replication occurs. And DNA replication is a complicated process that we are going to discuss during Unit 4. And G2 is when the necessary proteins for division are synthesized. Checkpoints between each of these phases ensure that everything goes smoothly and that there are no errors. So again, let's look at this. After mitosis, we get two cells, as you can see here, right? Let's look at this one cell over here. This one cell goes into G1, it grows, it does its functions, yada yada yada, everything is fine, until it gets to the S phase. During the S phase, it's going to duplicate its genetic material. As you can see here, we have two unduplicated chromosomes. Now here, we have two duplicated chromosomes. Moving on into G2. That is when it's going to create all the proteins that it needs to begin dividing. It's going to get into mitosis and the several stages of mitosis that we're just about to get a closer look at. It engages in cytokinesis, if it will at all. And then you have your product. And it can go back into G1 or G0. The important thing about G0 that you need to understand is that that is cell cycle arrest, as you can see here. G0 or G0 basically means that the cell is not going to divide any further. It means that that cell can no longer do it or will no longer do it. That's the last cell of the cycle. It will not divide any further. That is G0. That is what that means. Now, there are 4 stages of mitosis that we're just about to get a closer look at. There is prophase, metaphase, anaphase, telophase. There is a good mnemonic device that I always use to remember this. It's called PMAT. PMAT. It's in order. P-M-A-T. Prophase, metaphase, anaphase, telophase. That is the order of the stages in mitosis. Cytokinesis, if it occurs, will happen simultaneously toward the end of mitosis. Now, this is important, okay? If it occurs, this is a reminder that cytokinesis does not need to happen in order for mitosis to occur. Let's give you a throwback to biodiversity. As I always say, if you have not taken biodiversity, this does not affect you at all. But if you have, this may be important information to know. If you remember Plasmodium in biodiversity, this was a huge cell with many nuclei. We're talking about millions, thousands, hundreds of thousands of nuclei, okay? But the organism was still one single cell that never divided. It is just one huge cell with many nuclei inside that cell. How did those nuclei divide? How did we get so many nuclei? We got it through mitosis without cytokinesis. That is only one example where cytokinesis did not happen. And it is the proof that it is not necessary in order for mitosis to occur. Mitosis. What you start with is what you end with. That is the motto of mitosis. Everything that you begin with in regards of genetic material is what you are going to end with. If you began with a diploid cell, you will end with 2 diploid cells that have the identical genetic material that the parent cell had. Mitosis is used by cells for growth and repair, and that is self-explanatory pretty much, because every cell that comes through mitosis is going to be the same as the cells that came before mitosis. It takes place only in eukaryotic cells. In mitosis, duplicated chromosomes are separated into 2 identical cells. Perfect. That's easy to understand. Let me go back a little bit to this one right here. Only in eukaryotic cells. You might be thinking, well, I know for a fact that bacteria divide, right? Well, they don't do it through mitosis. The reason for that is because in order to do mitosis, you need several chromosomes. Bacteria do not have chromosomes. Bacteria have a single loop of DNA. So they engage in a somewhat similar process that we call binary fission, okay? But I just wanted to mention that in case you were wondering. Mitosis is characteristic of eukaryotic cells only. Now, the number of chromosomes per cell stays the same, and that will be the same for all mitotic divisions, okay? If you began with 46 chromosomes, as we humans do, after mitosis, you're going to have two cells with 46 chromosomes, exactly the same. Step 1 is prophase. During prophase, the chromosomes appear condensed and the nuclear envelope is not apparent. So, the nuclear envelope disappears. That is what this means. You know we have a nucleus in eukaryotic cells, but when mitosis is about to occur, it begins by getting rid of that nucleus. So for once, the genetic material is over the cytoplasm. But that genetic material, after it has already been replicated. Remember that we are looking at the beginning of mitosis. Step 1 happens after G2. And what happened before G2? We had the S phase, where genetic material was duplicated. So at this point, we already have twice as much genetic material as we are meant to have. So what's going to happen is that genetic material is going to be equally separated in two new cells. The mitotic spindle, made from microtubules, forms from the two centrosomes and attaches at the kinetochores. That is the first step in mitosis, prophase. Note the microtubules from one centrosome connect to one side of the kinetochore, while the opposite centrosomes, microtubules, connect to the other side of the kinetochore. The centrosomes serve to organize the microtubules and move toward opposite sides of the cell. And yes, this is what the microtubules are going to be for. That is why we have a kinetochore. We are going to see more the importance of this as we move through the other stages. This is a small demonstration of what prophase is like. You already have condensed chromatin into chromosomes, the nuclear envelope disappears, and the centrosomes begin to move toward opposite ends of the cell, at the same time that the microtubules begin to attach at the kinetochores of each duplicated chromosome. Step 2 is metaphase. Chromosomes are directed by the spindle microtubules to line up at the metaphase plate. This is an imaginary plane between the spindles. It is comparable to the equator on Earth. It is not a structure, it is just a line that represents the center. That is the metaphase plate. It is that area in between the cell that we just happen to call metaphase plate. And that's all metaphase really is. It is just the lining up of the chromosomes on that area, that equator area. Note, all chromosomes will line up on the metaphase plate during mitosis, as you can see here. This is what we refer to as metaphase. They are lining up in the metaphase plate, which again is just this imaginary line going through the center of the cell, okay? This is what we're looking at here. The microtubules are making this happen. The microtubules are already attached to the kinetochores, and they are aligning the chromosomes in the metaphase plate. The third step is anaphase. This is where the chromosomes begin to get separated. Chromosomes are pulled apart by the spindle apparatus towards opposite poles. the chromatids are now separated and are considered separate chromosomes. So, it is important that you look at this video right here. Here, the chromosomes have already been separated slightly, and now they're being pulled apart. But this is what I want you to notice. Right there, before the chromosomes were separated from the centromere, they were considered one chromosome. Now that they are fully separated, they are two chromosomes. Okay? Remember, this is one chromosome. This is two chromosomes. Alright? Step 4. Telophase. The nuclear envelope reforms around each set of chromosomes, and chromosomes uncoil back into their chromatin form, back into their uncondensed form. Remember, chromatin and chromosomes are all made up of the same genetic material. The only difference is the arrangement. If it's a chromosome, it's pretty tightly coiled. If it is chromatin, it is not so tightly coiled, but it is the same genetic material. All of it is just DNA. Cytokinesis will usually take place during this step. During telophase, cytokinesis will take place if it's going to happen. And yes, that's the general convention. That's what usually happens. But usually happens is not the same as always happens, okay? So cytokinesis is independent of mitosis. Cytokinesis is not a part of mitosis. So here we're looking at telophase together with cytokinesis. It could very well be without cytokinesis. We can see a cleavage furrow. This is what we call right here. Cleavage furrow is when these begin to separate slightly, and then there is this structure called the contractile ring that only gets smaller, 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 smaller until it divides both cells in two. And after the cells have been divided, we know already what's going to happen. A nuclear envelope will form for this new cell, and the new nuclear envelope will form for this other cell. And the chromosomes are going to uncoil, and they are going to become chromatin. As you can see here, we get a new nuclear envelope, and the chromosomes pretty much disappeared because they become chromatin, which is not so easily visible. Cytokinesis, separation of the cytoplasm of cells. That is the process of cytokinesis. It is just the separation of the cytoplasm. And this includes organelles too, okay? In an animal cell, a ring will form around the center of the cell, and a groove called the cleavage furrow will appear. The cell will pinch into two new cells. Now, this is able to occur because, remember, animal cells do not have a cell wall. A cell wall is hard. It is not easily broken, okay? So an animal cell is able to just divide into two new cells, and that's that. However, a plant cell can also do mitosis, and it can also do cytokinesis. In a plant cell, a cell plate is synthesized between two cells when vesicles filled with cellulose move to the midline of the cell. That is cytokinesis in plant cells. The newly formed cell plate gives rise to one side of the plant cell's wall. So this is what we're talking about. Vesicles filled of cellulose come in between the two new nuclei. They release the cellulose, and this new cell plate appears. Eventually, that new cell plate becomes a new cell wall, and you have two new plant cells. Where you had one, now you have two. So this is a summary of mitosis. We begin at interphase, which, as I told you before, it's merely the cell whenever it's not in mitosis. We know there are 3 steps here, right? There is G1, the cell is growing. S phase, DNA is synthesized, it is replicated or duplicated, and then the G2 phase is where these proteins necessary for division begin to appear, they begin to be synthesized. From G2, then we move into mitosis. And we begin with step 1, prophase. The nuclear envelope disappears and chromatin becomes chromosomes. Next, we go into metaphase. During metaphase, the microtubules have already attached at the kinetochores of each duplicated chromosome, and during metaphase, those microtubules are going to make them line up through the metaphase plate. Then, in anaphase, the microtubules will pull apart on the chromosomes. They are pulling on different directions. So, one duplicated chromosome, as you can see here, becomes 2 unduplicated chromosomes. One duplicated chromosome becomes 2 unduplicated chromosomes. And lastly, during telophase, one cell becomes 2 if cytokinesis is happening. Yes, telophase and cytokinesis. One cell becomes 2, pinches in the middle using the contractile ring, makes a cleavage furrow, and during telophase, a new nuclear envelope appears, and the chromosomes relapse back into chromatin. Not shown in this picture, by the way. But that's what happens. Cytokinesis, again, is an independent, simultaneous process that occurs towards the end of mitosis. So that's mitosis, guys. We're gonna go over the review questions, and we will see if you can answer them before I show them on the screen. So how many chromosomes are in this cell? First of all, we have 1 chromosome, 2 chromosomes, 3 chromosomes, 4 chromosomes. Alright. Are they duplicated or unduplicated? Well, we can see that we have chromatids. They are duplicated chromosomes. What is the ploidy of this cell? This one might be tricky if you do not correctly remember the meaning of ploidy. Okay, let's see. It is diploid. Now, you could easily answer this by looking at the colors. Sure. Color red, color blue, 2 sets, diploid. But also notice that we have big A's, and we have little a's. These represent alleles of the same gene, don't they? That is all they are showing. Remember, they're in the end they are all A's, so they are the same gene, absolutely. But one is big, one is small. That represents different alleles. We could say the A is for hair color, and this other A is also for hair color, but they are different colors, okay? How many chromatids are present in this cell? You need to know the difference between chromatids and chromosomes. The answer here is 8. We have 4 duplicated chromosomes. For each duplicated chromosomes, we have 2 chromatids, so that means we have 8. How many chromosomes are present in this cell? Do not say 1.5. We have 3 chromosomes, okay? We have 1, we have 2, and we have 3. And they are unduplicated chromosomes. Clearly, because there is no centromere anywhere in here. There is no identical chromosome. What part of interphase does this process occur in? So notice, we had 1 chromosome. We still have 1 chromosome here, but now it's duplicated. We had 1 chromosome, and we still have 1 chromosome here. Also duplicated. DNA replication occurred. When in interphase does this happen? It occurs during the S phase. DNA replication. Please, again, keep in mind, there are 2 chromosomes in this cell. There are still only 2 chromosomes in this other cell. And that's it, guys. Please remember to do the quiz so that you can get your 2 PPP points. And I will be posting this presentation, the PowerPoint presentation, online to make it easier for you to fill out the follow-along sheet as well. Hopefully you were filling it out as we were doing the lecture portion, but if not, I will be posting this PowerPoint presentation after I post the lecture. Thank you and have a nice week.